Here we're going to integrate all the concepts that we've learned all this semester and maybe just add a few more things. First, we're going to start with what is a species, or what do we call species? And let's see how we name species. Species names have two parts. The first part is the genus, which is always capitalized, and the second part is the species, specific. Both together are the species name. So whenever you have a species, you have two parts, the genus part and the specific species part. And sometimes you can see the genus is abbreviated where only the first letter is kept and then the species name is spelled out. This is the species name for a squirrel. You can have the species name of a dog, which is Canis lupus or Canis domesticus. Canis is the genus name, domesticus is the species name, although you will always have both together. So you will say Canis domesticus, and then if you're having wolves to separate, you will call them Canis lupus. So they have the same genus, but they're different species. And Linnaeus was the one who invented this naming system. You can have species within the same genus, so they share the same genus, like the example of dogs and wolves. And that would be species that are very closely related, that are within the same genus. And he kept grouping them by how closely related the species were. So we start with the species name. Here the squirrel, in this case, is Cirrus carolinensis. Then you have all the species that are in the genus Cirrus, and then all the different genera that are closely related are within a family. Genera is the plural for genus. So all those groups form a family. Then families of species that are closely related are grouped together into orders. Orders are then grouped together into classes. So this squirrel belongs to the genus Cirrus, to the family Cirridae, to the order Rodentia, which is with other rodents. It includes mice and chipmunks and other rodents. Then it goes to class. The class Mammalia is a mammal, and then subphylum is a vertebrate, and phylum Chordata, so it's a chordate, to the animal kingdom in the domain Eukaryotes. So you keep having larger and larger groupings of species. So it's a hierarchical system that in some way tries to capture how closely related the species are. And in modern times, we use phylogenetics to look at the relationships of species or groups of organisms. Here we have a phylogeny of apes. We have chimp, humans, gorillas, orangutans, and gibbons. Humans and chimps share the most recent common ancestor. The ancestors are, and the nodes, they are the most recent ones to split. If you go back in time, you have a common ancestor between gorillas, chimps, and humans. And then as you keep going back in time, you eventually find a common ancestor of all the apes. So notice that as we go deeper into the tree, that's when we go back into the different ancestors. The order of the groups here at the top doesn't really have any meaning. So it's not that if those on the left are less evolved than those on the right. Actually, there is no such a thing. The ancestors are when you look deeper into the branches of the tree. That's when you see the ancestors. All the organisms listed at the top of the tree, all those in the leaves, so to speak, those are all of the same time. Those are all existing lineages that are present today, that have the same evolutionary history, that have been evolving for the same amount of time. They all trace back to the very first living organism on Earth, and they all are the result of billions of years of evolution. So even if you have this tree and you keep adding other animals, and eventually you will add other organisms like plants and even protists and bacteria, all the existing organisms in the tree of life all have been evolving for the same amount of time, all are equally evolved. They might be different and different in complexities, but they all have been evolving for the same amount of time. It's when you look deeper into the branches of the tree, that's when you look at the ancestors. And phylogenies can be depicted in different ways. like the one we saw, you can rotate the branches and the meaning will still be the same. So here we have humans next to gibbons, but that hasn't changed the meaning of the tree because what matters is the pattern of the branches. In this tree, humans and chimps are still the most closely related. The information hasn't changed. They are the ones that share the most recent branch, just like here. But the order here does not have any meaning. So even though it would appear that gibbons and humans are now more closely than they were over here, the order in this on the top doesn't really mean anything. You have to look at the pattern of the branches, and the pattern of the branches is still the same. Humans are most closely related to chimps. Human and chimp together are more closely related to gorillas. And then gorillas, chimps, and humans share a most recent common ancestor with orangutans. And then all those together share an ancestor with gibbons, which is exactly the same information as here. And you can also put the tree sideways and still has the same information. So think of phylogenies kind of like how you think of a family tree, where all the organisms at the top are the latest generation. So you and your siblings will be there, and the most recent common ancestor between you and your siblings is your parents. And then you have your cousins, and the most recent common ancestor you share with you, your siblings, and your cousins are your grandparents. And then you have your second cousins, and the most recent common ancestor you share with them are your great-grandparents, will be over here. And this doesn't mean that you are more evolved than your second cousins, or that your second cousins are more ancestral than you or your siblings. All the organisms in this existing generation are equally as evolved. And I'm not saying that you're more evolved than your parents or grandparents, but they are your ancestors. So as you go down in the tree, that's when you start going back in time into the previous generations, and eventually you go deep enough into species that were the ancestors of the existing species. So when you look at an evolutionary tree, and we're looking at humans and another group of mammals, the thing that joins us is the common ancestor of all mammals. And then we're looking at humans, mammals, and amphibians, like frogs, we will share a common ancestor with them, which was the most recent common ancestor of all four-legged vertebrates or tetrapods. And if you keep going back in time, you will find the most recent common ancestor we share with fish, and so on. As you keep going down in the phylogeny, you will find a most recent common ancestor we share with any other living organism. And the way these phylogenies are constructed is based on the traits that the species have in common. So we will produce a table like this that looks at the different traits, and then the list of organisms. Ones symbolize the presence of the trait, zeros the absence of the trait. If they have a lot of those traits in common, like here humans and gorillas have a lot of those traits in common, the more traits you have in common, that means the more closely related you will be in the phylogeny. And phylogenies used to be made mostly of anatomical data because that was, or morphological data, because that was the data that was more available at the time. But as molecular techniques have become very common, now most phylogenies are based on DNA or genomic data. And there are specific sequences that have low mutation rates that are the most commonly used in order to determine phylogenies. It is important to look at what trait you're using for your phylogeny, because you want to use traits that are the result of common descent. And these traits that are the result of common descent are called homologous. That means traits that were inherited from the most recent common ancestor of those groups. The opposite of that are analogous traits. Those are traits that appear independently on the two groups but are not the result of common ancestry. So you want to look at traits that do reflect common ancestry versus looking at traits that don't really give you information about common ancestry. Homologous traits are traits that have the same evolutionary origin, like the arms of these different mammals. They all come from the forelimb of the original mammalian ancestor. They have been modified in the different groups for the different purposes, but if you look at the bones present are the same, and you can find equivalences across all these different types of forelimbs. On the other hand, an analogous trait is one that evolved independently on each of these groups that doesn't reflect common descent. So these types of traits are not informative when we're making phylogenies. Examples of that are the wings of an insect with the wings of a bat and the wings of a bird and the wings of some other flying vertebrate. Those are not homologous. They did not inherit those wings from a common ancestor. There was no common ancestor with wings between bats and birds. They evolved those traits independently. So analogous traits are not informative when we're trying to make phylogenies. Another concept that is important for phylogenies is the concept of parsimony. This means the least number of steps in order to group those organisms. As you look at the different groups and the traits that they have, some organisms acquire new traits and some organisms have lost traits. For instance, we acquire the trait of being able to walk in 2 feet, but we lost traits like having hair and we lost traits like having a tail. Monkeys have tails, but apes don't have tails, so we lost that trait. Parsimony looks at those instances in which new traits appear or traits disappear and tries to come up with a solution that involves the least amount of steps. And the idea is that is the most likely scenario to have happened, is the one that involved the least number of steps. So in this case, the red color, if you put these two groups as separate, you're saying this appear here and then this appear here. That means there are 2 steps, 2 appearances of a trait. Versus saying the trait appear in the common ancestor of these 2 groups, that's just 1 appearance, and that means fewer steps, a more likely scenario that that's the way it happened. So we look at traits that are the result of common ancestry in order to form these phylogenies. Here we have a phylogeny of all the chordates, including tetrapods. Mammals will be here, part of this. Reptiles, birds, amphibians, fish, and other non-vertebrate chordates. An example of an homologous trait, 4 tetrapods. Tetrapods are those with 4 limbs. Amphibians, like frogs, will have 4 legs. Mammals have 4 legs. Reptiles have 4 legs. Birds, they have 4 legs. It's just 2 turn into wings and 2 are still legs. All these are called tetrapods. Homologous traits for all these groups are having a vertebral column. Everybody here in this group does. Having 5 digits. Originally, everybody here descended from an ancestor with 5 digits. And an analogous trait would be looking at mammals and birds, which both have endothermy, both have the ability to regulate temperature, but they did not inherit that from a common ancestor. What kind of trait will you call the presence of wings in bats and in birds? Now let's look at how species emerge. And species appear from an original population that had genetic variation in it. And for some reason, the population got divided, either because they were separated by a geographical barrier, or they started inhabiting different ecological niches while still on the same location. And over time, as those species became reproductively isolated, their allele frequencies become more and more different until they got to a point where the populations are reproductively isolated and now they are considered 2 different species. We used to think that most speciation happened allopatrically, that means in different locations, like this species of anoles, these lizards living in different islands. Originally, they started from one population that split into each of these different islands, and while they were separated in each of their islands, they evolved into separate species. And that's how we thought most of the speciation happened, by being geographically isolated from one another. But we're seeing more and more cases of sympatric speciation. So these are species that appear while they still live in the same area, even in the same forest. Like the examples of the lizards within one island. They specialize on different parts of the canopy, and they have become different species as they adapted to the specific niche that they inhabit. And now they can no longer interbreed with each other even though they live in the same forest. And we also talked about evolution without speciation. So you have evolution with speciation. It's called macroevolution. This is what led to the diversity of species today. But we also have microevolution, and this is when evolution happens within one species. And this is happening all the time in pretty much all species, unless those populations that are under Hardy-Weinberg equilibrium, which is very rare. The allele frequencies on a population are changing, either because mutations are happening, organisms are migrating in and out of the population. You have genetic drift, random events that are changing the allele frequencies of the population. You have different preferences in mating, so that mating is not always random. You have natural selection, favoring certain alleles over others. So in most cases, any population that you will look at will have microevolution happening. And then eventually, in some cases, that might lead to the separation of different species. So just the key to remember here, evolution is the change in allele frequencies. And this can happen by any of these factors, by random events in genetic drift, by natural selection favoring some alleles over others for that specific environment, migration or gene flow, organisms moving from one population to another, and mutation. This is how different alleles originate, by one allele mutating and forming a new allele that didn't exist before. So how do new alleles appear? And what is an allele first? So when we have a trait like eye color, we'll say the gene for eye color, then the allele is the specific character for that trait. Is the eye color blue, brown? What is that trait? That is the allele. Blood type, so blood type will be the gene for blood type. And then the allele is it blood type A or blood type B? Or if you're looking at the Rh factor, is Rh positive or is Rh negative? So we have genes that code for traits, but what the trait specifically is, what version of that trait you're getting, that depends on the allele, which is the specific DNA sequence that is in that region. And that specific sequence is going to produce a particular protein. So if you have the allele for blue eyes, that's going to produce a protein that will give the blue color. The sequence in that DNA produces a protein that gives you the blue color, while the allele for brown eyes has a sequence of the DNA that, when it's transcribed and translated into protein, produces a protein that gives you the brown color. So in that, where will the mutations happen that will create new alleles? And we know within a cell, cells use their DNA to transcribe it into RNA and use that RNA to translate it to make proteins. This is how the cells do their day-to-day life. They need proteins in order to do their day-to-day activities. But mutations that happen in this process would only have an effect in that particular cell at that particular time. So if you have a mutation in the RNA while you're transcribing the RNA, that mutation might result in a defective protein, but that is only one RNA sequence of the many RNA sequences that cell is producing, and that will last a very short time, and it's only in that one cell compared to all the millions of cells you have in your body. So that one mutation is not going to make any real difference. For mutations to really occur, a mutation that actually has an effect are mutations that happen in the DNA. So we talked about the DNA being the recipe book and the RNA being a photocopy that you make, a temporary photocopy, so that you can make the recipe, and the ultimate recipe is the protein. As long as your original recipe book is okay, then if you make a mistake in one of the photocopies, it's only going to change the outcome for one time you make that recipe, but it's not going to be a permanent change. But if the mistake happens when DNA replication occurs, DNA replication is when the cell is going to divide into two cells and it has to give an entire copy of its genome to each of the daughter cells. If a mistake is passed to the daughter cells so that the recipe is wrong in this cookbook, anytime the cell tries to make that recipe is going to have that mistake. So that's what we call a mutation. It's something that is permanent, that gets passed on from one cell to another. A mistake in the RNA is temporary, even within that cell. It's temporary because the cell is making RNA all the time. Those recipes are being transcribed all the time. So if one copy out of thousands has a mistake, it's really not a big deal. And that RNA won't pass to the offspring. That RNA is just used to make proteins. The DNA is the original copy, and that is the one that will be used as a template to duplicate all the genome and pass it on to the daughter cells. So before a cell divides, it needs to replicate its DNA. And every chromosome on the cell starts like this, a double-stranded DNA, will have to be replicated. So the first step in that process is to unwind the strand, and that unwinded strand then gets duplicated. A complementary strand is synthesized for each of the strands, and now you have two complete DNA molecules, and these complete DNA molecules are now two sister chromatids that, as long as DNA replication went without any issues, they should be identical. But if there was a mistake in the original DNA template, or a mistake happened during the DNA replication process, then those mistakes, if not corrected, will be passed on to the daughter cells and will be permanent. So the first step when a cell is going to divide is to replicate its DNA, and that's what happened here. We go from having a single chromatid to now cells with sister chromatids. And then in mitosis, if that cell is going to divide by mitosis, the sister chromatids will be separated so that each chromatid goes to one of the daughter cells. If there was a mistake in the original chromosomes, when those chromosomes were replicated, if that mistake wasn't fixed, then that mistake will be replicated, and that mistake will be present in the daughter cells. Or if a mistake happened during DNA replication and it goes uncorrected and the cell divides, then that mistake will be present in the daughter cells. So what type of mistakes could happen? This mistakes is what we call mutations, although when we say mistakes, we assume they're bad. Mutations are not necessarily bad. They could be neutral most of the time. In some cases, they're bad, and in some cases, there might be improvements. So those changes could be an insertion, inserting a nucleotide that wasn't present, and that would change the sequence from thereafter. It could be a substitution. One base pair is changed for a different base pair, and what effect it will have depends on what that codes for. Or it could be a deletion where a base pair is removed, and then it would change not just that particular part, but also how you read the rest of the sequence from there on. An example of a substitution is what's called sickle cell anemia. We have the original DNA sequence, and this adenine was substituted by a thymine. And when the RNA is read, it's read in groups of 3. Those are the codons. This group of 3 codes for the amino acid glutamate. Once you change that A for a T, now this group of T codes for the amino acid valine, and they are amino acids of very different proteins. Glutamate is polar, valine is non-polar. When the protein forms and starts to fold, it folds in a very different way because the tertiary structure of the protein depends on the interactions between polar and non-polar amino acids. And if you change a polar for a non-polar, those interactions will be different, the tertiary structure will be different, and that results in this agglomeration of the hemoglobin protein that then changes the shape of the red blood cells, and that makes them difficult to navigate to the blood vessels, and that's what causes anemia. But most of the time, mutations don't have an effect, and the reason for that is that the majority of the genome doesn't really code for protein. Most of the genome is not transcribed at all, and the parts that are transcribed, not all of them are translated into protein. So if you look at all of your genome, the majority of their genome are transposable elements and repeated DNA. That most likely comes from viruses that inserted their DNA into our DNA. Same thing with this repetitive DNA that is not transposable elements. Then we have some unique non-coding DNAs. There are the known regions that we know that are either introns, so those are sequences that are transcribed but not translated, and other regulatory sequences like the promoter regions or regions where activator proteins bind, that's just enhancer regions. They can also be parts of the DNA that are transcribed into RNA for RNA of interference. They won't be translated into protein, but that RNA of interference can regulate the expression of other RNA. And then a very small fraction of the entire genome, just 1.5% of the genome, is actually expressed. Are either genes that code for protein or genes to make ribosomal RNA, transfer RNA, RNA that has specific enzymatic functions in the cell. So of all your genome, really only 1.5% of it is what has information for proteins and for physiological function. The majority of it has unknown function, and a fraction of that is for regulatory purposes. And when you compare the genome of different species, you find that there's a lot of similarities. And there is families of genes that are present across all the different species. So does it matter in which cell a mutation happens? Is it the same whatever cell gets a mutation, or does it matter which is the cell that is getting a mutation? So if you think of how we get to be who we are, we all started as an egg and a sperm that fused together and combined their chromosomes. So we started as that one original cell. And then that cell divided by mitosis many times to give rise to the embryo and then the baby. Those cells kept dividing by mitosis to give rise to who we are today. If a mistake happened very early on in one of the few cells that were doing mitosis here, even if it's just one cell that has a mutation, since those cells would give rise to a large part of the organism, they could have a very big impact, as those cells will continue to divide to produce a large set of the organs or the tissues in that organism. On the other hand, if a cell mutates now in a grown-up person, that mistake is very unlikely to have an effect. Most of the time, the cell will commit apoptosis if it cannot fix the mistake, or other cells will force it to do apoptosis. Or in the worst case scenario, that cell might become cancerous, and that will be a problem. But those mistakes will not be inherited in the next generation. What makes a mutation heritable? Where does it need to happen in order to be passed on to the next generation? The next generation starts as that cell that is the result of the egg and the sperm fusing together. So in order for a mutation to be passed on to the next generation, it has to occur in a cell that will eventually give rise to either eggs or sperm. And the only cells that produce gametes, which are the cells that will be passed on to the next generation, are the cells in the gonads, the testes for males and the ovaries for females. Those cells are called germline cells, and those are the cells that will divide by meiosis to produce the gametes that will have the information that will be passed on to the offspring. So mutations that happen anywhere else in the body will not be inherited by the offspring. Only if a mutation happens in a cell that will eventually give rise to the sperm or eggs, those mutations will be passed on to the offspring. Those are the only cells that are doing meiosis. All the other cells are dividing by mitosis and are producing diploid cells that will not be passed on to the offspring. Another thing that matters is what type of gene is the one that has the mutation. If you have a gene that codes for a specific trait, like eye color, okay, maybe instead of having green eyes, you have hazel eyes. It's a slight change. It's not really a big change. But if it's the gene that coordinates the development of eyes, then you might have eyes or no eyes. That is a very big change. Or in this example of these fruit flies normally only have one pair of wings. But here, the gene that starts the development of the wings was activated twice, and now this fruit fly has two sets of wings, which is a very different phenotype. The process of development needs to be a very controlled process. We start with stem cells in the embryo that can develop into any tissue in the body. And that process of how they differentiate into each specific tissue has to be very carefully controlled. This is a process that occurs in all multicellular organisms, animals and plants. They all start as one single cell, and that cell divides by mitosis many times, giving rise to more and more cells. And as that process happens, changes in the embryo start happening so that they determine which parts of the body of the organism they're going to create. And how those decisions are made are basically responding to signals on the external environment, either from the neighboring cells or cells that are farther away, that are activating signal transduction pathways that ultimately change gene expression in each of those cells. As we look at an embryo throughout development, we see very characteristic patterns of gene expression that change through time. So this is one embryo of a fruit fly. This is the anterior part of the embryo, the posterior part of the embryo. And this is, as it goes through time, you see a very specific pattern of gene expression that changes over time in the embryo. And these genes that are activated this very early on in the embryo are called developmental genes. And what they are doing is they are setting on march, they're activating this set of genes that would then produce more specific structures. So they are kind of like an orchestra director. They are setting on motion the development of the future structures. And those developmental genes are very conserved. When you look at organisms as far away as this nematode worm or fruit fly or a chordate and humans, you see, look at the colors, it's telling you what are the genes found and where are they expressed. And the pattern of expression of which are more expressed towards the head and then which is towards the tail is very conserved in all these different groups of organisms. But what happens is, what are the genes that those developmental genes then activate later on? So let's look at this example in tetrapods. We have humans, birds, and a common ancestor of both groups. And these are 5 genes that are involved in the development of limbs. In humans, this developmental gene called TBX5 activates these first 2 genes and this last one here. And that results in the formation of an arm. The same developmental gene, TBX5, in birds activates the first gene, just as it does in humans, but instead of activating the second, it activates the third and fourth, and it doesn't activate the fifth. And as a result, you have a wing. So these developmental genes, like I said, tend to act as transcription factors that go and activate other genes. And usually the change happens in which are the genes that they activate, while the developmental gene itself tends to be very conserved. And this might explain the patterns we see in evolution. So some propose this gradualism model and some propose this punctuated equilibrium model. We see examples of both in nature. The gradualism is that changes happen very slowly and there are very small changes that eventually add up to big changes, while the punctuated equilibrium model says there are some big changes and then they stabilize and they stay in that form for a while, and then you have another big change happening. These two are possible if you look at what are the genes that are being modified. If the genes modified are very specific traits, like we talk about specifically the color of the eyes, then you have very small changes that are building up over time. But if the change is big, either you have a new set of eyes and now you have, instead of having 2 eyes, you have 4, that's a huge change. And that's just a change in one gene that controls the development of eyes. So it's a gene that acts very early in development, will have very big changes that would result in a big evolutionary jump that might be beneficial and might stay and continue in that group. So what are those changes caused by developmental genes? So again, looking at what genes are just sections of DNA that code for specific proteins. In order to code for a protein, they have to be transcribed into RNA and then translated into protein. Developmental genes are determining which genes are expressed where and when. And how they do that is by manipulating any of the steps in gene expression that we know of. So they can start from changing the structure of the chromatin or working as transcription factors to make transcription possible, changing the way the RNA is processed so the different introns or exons are added or removed, whether the RNA is going to be exported out of the nucleus, and how long it might be able to be translated for, or if it's going to be broken down. and how long the protein that it's produced is going to be active. The most common known mechanisms for gene expression during development, control of gene expression, are such as epigenetics, the tightening of the chromatin. A cell that is going to become a neuron will not have to express genes that are only present in muscle or in kidneys or in the liver. So those genes that will not be expressed in a brain cell will be silenced by tightening the chromatin, making the chromatin very tight so that it's not going to be transcribed. While the genes that are necessary for that cell will then be looser so that they can be transcribed. Most developmental genes act as transcription factors, and they might activate then other genes that are also transcription factors, so they activate gene expression by coding for proteins that will then act as transcription factors that will start the expression of genes that produce proteins for a more specific function. Or they can also be regulator proteins like activators or repressors. We can also have effects in post-transcription regulation, especially in the editing of the RNA. We talked about the process of alternative splicing, where the same RNA transcript is edited in different ways, with some pieces being removed, and those will be called introns, and other pieces being left, and those are called exons. And different cells, or cells at different times, can remove different introns and assemble different exons so that the resulting proteins will be different. And this might explain why, although we only have 25,000 genes in our genome, it was estimated it was going to be much more than that. And actually that number keeps dropping. The recent estimates are that there might be just 20,000 genes, and some people think it might be even lower than that. But we have more than 100,000 different proteins that are being made, and that's what we call the proteome. So the genome is the number of genes that we have, and the proteome is the number of proteins that we produce. And some estimates say the number of proteins might be up to 500,000, so 500,000 proteins, and only about 20,000 genes. It's a big difference, and the reason why that can happen is because the same gene can be transcribed and then edited in different ways by alternative splicing, producing different types of proteins. So based on that, what do you think is bigger in terms of number of types? Number of genes in the genome, number of RNA transcripts in the transcriptome, or number of proteins being produced in the proteome? So with this, I hope to give you an overview on how the processes that we've studied this semester all come together to give rise to the diversity of organisms and the diversity of cells in our own organism, and how those changes are orchestrated, starting with one single cell that then develops into a full multicellular organism.